So after having the knowledge of these different protein structures, I want to discuss how the protein structures can be altered. So in this video, I will discuss hydrolysis of proteins to break up the protein sequences, and denaturation of proteins to break up the interactions that makes the three-dimensional shapes of the proteins. So first we'll see what is protein hydrolysis. The hydrolysis is the opposite reaction of the peptide bond formation. So in protein hydrolysis, the peptide bonds are broken through a hydrolysis reaction. So this is a natural reaction that happens in our stomach. When we consume food that contains proteins, so we have enzymes in our digestive system that can hydrolyze these proteins. So they basically catalyze the hydrolysis of proteins to produce the amino acids. So it breaks up the amide bonds. Specifically, they break up the amide bonds to gain the single amino acids that was made the primary structure. So I have an example here. A tripeptide, alanine glycine serine. The tripeptide can hydrolyze to obtain the three amino acid residues. So in the protein hydrolysis, basically this amide bond is broken down. At the same time, it will just add a water molecule to both sides to gain the single amino acids. Now, once this tripeptide is broken down, it will result with the same amino acids that was made up of those tripeptides, alanine, glycine, and serine. So protein hydrolysis is the only way that can break up the amide bonds to give these single amino acids, and that happens in our body. Now we will see what is denaturation of proteins. So this is different from hydrolysis. Denaturation of protein is the disruption of the interactions that was maintaining the secondary, tertiary, and quaternary structures of proteins. And you need to remember that this denaturation does not affect the amide bonds at all, because amide bonds are very strong covalent bonds, and that can be done only by hydrolysis of proteins. Denaturation is a different process that happens due to multiple factors, and in denaturation can happen by increasing the temperature or making the pH of the medium. It can be acidic or basic, and also adding some chemicals or organic compounds or heavy metal ions to the protein. It can denature, and also adding a mechanical agitation. So this will just disrupt the interactions like hydrogen bonds, hydrophobic interactions, or salt bridges can be altered by due to these factors. And when the interactions are disrupted, what happens? Those globular proteins or the three-dimensional structures will of the proteins will destroy, and proteins can unfold to give a simple polypeptide chain. So what happens when the proteins are denatured? Now, at the beginning of this lecture series, I mentioned different proteins have their own different functions, right? So the protein structure is directly correlated to its function. So if the structure is altered, that protein cannot form or cannot function uh, to give cannot function anymore. So those proteins will no longer biologically active. Okay, now we will see how these factors are affecting the protein structure one by one. Now, what is the effect from the heat? So the proteins will generally denature when those are heated above 50 degrees of Celsius. When the temperature is too high, so that heat can disrupt the hydrogen bonds. So this is the first thing to affect the hydrogen bonds. Because hydrogen bonds are not covalent bonds, or those are not very, very strong. Those are relatively strong, but not very, very strong. At when providing a high temperature, those hydrogen bonds can easily break up. What happens after that? When the hydrogen bonds are break up, we know that secondary structure of proteins, alpha helix or beta pleated sheets, are maintained due to these hydrogen bonds. So those can be unfolded. And also, in addition to hydrogen bonds, hydrophobic interactions are also affected. Um, so those interactions will be weak among nonpolar residues. And so if you just think about boiling an egg, so we know after boiling the egg, the texture of the egg is completely changed, right? So just like that, if when you cook meat, what happens? The texture is completely different due to this protein denaturation due to high heat. Um, but after heating, the texture will be changed, but it does not change the nutritional value of the proteins because the amino acid sequence is the same. So they are just making them more digestible, but the nutritional value of proteins will be the same. And high temperatures are also used to disinfect surgical instruments. So this is one reason that is used as a disinfectant method, because we know that disinfecting to get rid of bacteria and viruses. So these bacteria and viruses are also contains proteins to uh, make these germs. So basically, by disinfecting, the bacteria proteins, virus proteins are denatured or destroyed. So that's what the high temperature will do. So the second factor that affecting or that is used to denature proteins is acids and bases. So once an acid is added or a base is added, what happens? It changes the pH of the medium, right? The pH is a scale that is used to tell how much acidic is a medium is, or how much basic is a medium. Now, what happens when the acids or bases are added? So it will breaks the hydrogen bonds. Hydrogen bonds will affect the first, and at the same time it will disrupt the ionic bonds at salt bridges. Remember, the salt bridges are due to the interactions between positive ions and negative ions. When there are acids present, so these negative ions can protonate, or which means the negative ions. Uh, so, for example, salt bridges occurred in between these carboxylate ions and NH3 plus, for example, right? So what happens when we add so if suppose that this is a side chain of a protein and this is another side chain of another protein, the salt bridges occurred in between these negative and positive ions. So after adding um, an acid, for example, this negative ion will disappear due to attaching a proton, right? When H plus is when the medium is acidic, this O negative ion can um, just disappear due to protonating the hydroxyl charge. So there is no negative ion for the salt bridge to active, so the salt bridges will disappear. So these ionic interactions will disappear with an acidic medium or with low pH in the medium. Just like that, the acidity or basicity will affect the salt bridges. And so Acids and bases will denature proteins by affecting the hydrogen bonds and disrupting the ionic bonds. Now, this is one reason that tannic acid is a weak acid that is used in burn ointments, one ingredient in ointments that are used to treat burns. And what is the purpose of adding this weak acid to this ointment? Because once it's applied to the site of the burn, that will denature the protein around that wound. So it actually forms a protective cover and also prevents further loss of fluid from the burn because these proteins will coagulate in the site of the burn and it forms this protective cover. So for that reason, the weak acids are being used as a denaturing agent for proteins. So another denaturing agent is organic compounds. What are these organic compounds? More specifically, ethanol and isopropyl alcohol. We know that these are used as disinfecting agents, right? So their role is uh, to exchange the bacterial proteins hydrogen bonds. So the idea is to disinfect or get rid of the bacteria. So basically, those agents help to denature the proteins that contains in bacteria just by exchanging the hydrogen bonds to water with their own. So these bacterial proteins can make hydrogen bonds with water, right? So in place of the protein's hydrogen bonds, the alcohols will make hydrogen bonds with water. As a result, it disrupts the hydrogen bonds in proteins of the bacteria and that can change the structure of the proteins to denature it and it's not more active. And also it can disrupt the side chain intramolecular hydrogen bonding as well. Intramolecular means the hydrogen bonding that occurs in the same polypeptide chain in the proteins of the bacteria. So for the same reason, an alcohol swab is used to clean wounds or to prepare the skin for an injection because the alcohol passes through the cell walls and coagulates the proteins inside the bacteria. So this use of isopropyl alcohol or ethanol as a disinfecting agent is all about denaturing the proteins of bacteria. So that's how it cleans the skin before an injection. And denaturation of proteins by heavy metal ions. So the heavy metal ions specifically acting on the disulfide bonds of the proteins. The heavy metal ions are silver ions, lead ions, and mercury ion. So they can just disrupt these disulfide bonds or break up the disulfide bonds. I mentioned these disulfide bonds are strong covalent bonds that keeps the tertiary structure stable. Once these disulfide bonds are break up, they can no longer keep the shape of the protein, so proteins can deactivate. One application of these silver ions. So silver nitrate solutions are used in eyes of newborn babies to destroy the bacteria that cause gonorrhea. And agitation. So agitation is actually a mechanical method to denature proteins. For example, whipping of cream and the beating of egg whites are examples of mechanical agitation to denature proteins. So this just the whipping action will stretches the polypeptide chains until the stabilizing interactions are disrupted. So this is just to retain the desired texture. So what happens is just again denature the proteins without affecting the nutritional value. It's just change the texture of the protein. So proteins are denatured by agitation as well. So these are different methods of denaturation of proteins. And I have listed down all the denaturing agents and what type of bonds are disrupted and examples. So this is one of the major important table for you. You need to remember how these denaturing agents will affect and for what type of bonds those will be affected. So that will be tested in your exams and quizzes and in homework. Okay, so this is the end of this chapter. So you can try the homework problems and reach me if you have any questions.